A third of global crop yields is estimated to be lost due to pathogens and disease. In context with climate crisis, the decreasingly stable farming conditions, and an ever-growing demand for food, humanity is facing one of its biggest challenges. We will need to produce way more food while simultaneously limiting the environmental impact to ensure enough produce is available for all of us for generations to come. Reducing crop loss will need to be a major component of this, and significant efforts are needed for improved management of pests, including pathogens and weeds. Currently, the best tools against disease and pests are synthetic pesticides, yet these create a myriad of new challenges. Loss in biodiversity, deterioration of soil quality, and resistant pathogens are a direct consequence of synthetic pesticide use. In light of these challenges, we need to look for a better, more sustainable solution. In 2021, the citizens of Switzerland voted on whether to ban synthetic pesticide use for local crops. The results showed that there is a willingness for change, yet that there is a lack of effective and economically sensible solutions. This also became clear throughout our interviews with local Swiss farmers who reinforced this notion. One farmer told us, "If I didn't need to use synthetic pesticides, I wouldn't, that's logical. But the structure of my vineyard doesn't really allow me to give them up." Currently, there are 3 possible options to protect crops: synthetic pesticides, which themselves create more problems, GMO plants, which are currently illegal in Switzerland, and biopesticides, which are not as efficient as their synthetic counterparts. The goal of our project is to develop a novel approach to protect plants, which does not negatively impact the environment nor human health.
For the problem at hand, we came up with Omvs. The basic idea is to work with something that already exists, the plant's immune system. We used bacteria-originated outer membrane vesicles to prime the plant's immune system for an upcoming pathogen attack. We achieve this by expressing epitopes on the outer membrane vesicles, or short Omvs, that trigger the plant's immune receptors, the so-called PRRs, which then induces a plant innate immune response, the pattern-triggered immunity, therefore preparing them for future infections. The advantage of this strategy compared to conventional strategies is that by priming the plant for infections instead of killing off the pathogens, we minimize the selective pressure on the pathogens and hence prevent the rise in pathogen resistance. So, what are Omvs? Omvs are produced naturally by gram-negative bacteria and are used to transport biomolecules for communication, virulence, and many other purposes. Gram-negative bacteria possess 2 membranes, an inner and an outer. The Omvs are secreted by accumulation of phospholipids from the outside of the outer membrane to the inside, which causes bulging of the outer membrane and finally the budding of the Omvs. With this in mind, we decided to speak to Professor Leo Eberl, head of the microbiology department at the University of Zurich. He suggested looking at another type of vesicle, in addition to our Omvs, cytoplasmic membrane vesicles, or short CMVs, which are lysis-derived. You can trigger cell lysis by damaging the DNA via a mutagen like UV rays or mitomycin C, which activates prophages in the genome that induce the fragmentation of the cell into vesicles, known as CMVs. Key differences between Omvs and CMVs are the following. Omvs are produced via a natural mechanism of the bacteria and only contain periplasm, which is the space between the 2 membranes. CMVs, on the other hand, contain cytoplasm, and their production is based on cell lysis triggered by us. Once we had our idea set, we started designing our plasmids. Eventually, we came up with 3 main constructs. The first construct consists of expressing the immunogenic epitopes on the surface of the Omvs. For this, we use ClyA, a transmembrane protein with an extracellular domain to which we linked our elicitors FLAG22, alpha-18, or both. They are what's known as PAMPs, so they bind to the PRRs and thereby elicit an immune response. Therefore, as ClyA is integrated into the membrane, our epitopes are placed on the outside of the cell and eventually on the surface of our OMVs. While planning the first construct, we were confronted with a crucial question concerning the application method with which we want to induce an immune response. After several discussions with our supervisor, Professor Cyril Zipfel, one of the leading experts for plant immunity, we ended up with two possible courses: either spraying OMVs onto the plants directly, or releasing a genetically modified bacterium into the field that produces more OMVs once it senses a pathogen. Despite the strict GMO regulations in Switzerland, Professor Zipfel encouraged us not to limit our creativity and to pursue the latter as well. Taking his recommendation on board, we designed two simple constructs: one coding for a TEF protease and one for a Toll-B membrane protein with a TEF cutting site. Both constructs will be expressed in a Toll-B deletion strain, which lacks the Toll-B membrane protein that normally helps to hold the two membranes together. Thus, Toll-B deletion strain lacks outer membrane stability and therefore overproduces OMVs. The constructs work as follows. The promoter attached to the Toll-B gene is constitutively active, and thus Toll-B is produced. You see a wild-type phenotype. The Toll-B gene can be repressed by AHL, a quorum-sensing molecule secreted by bacteria. This results in the cease of Toll-B production. At the same time, the TEF protease gene is activated by AHL and starts to produce the TEF protease. This will result in two things. Firstly, the protease will cut the produced Toll-B at the inserted TEF cutting site, and secondly, no new Toll-B is produced since the gene is repressed. So in presence of AHL, we see the delta Toll-B mutant phenotype, which overproduces OMVs. And since AHL belongs to a family of quorum-sensing molecules, we could adapt our system to target a specific pathogen by using a quorum-sensing molecule that is unique to that pathogen. In theory, this inducible system could open up the possibility to release genetically modified commensal plant bacteria into the soil, where they would produce the immune-priming OMVs as soon as pathogens are sensed. Of course, we are aware that this part of our project would require further safety precautions, such as a kill switch, for example, and altogether is legally difficult to implement and/or even test in Switzerland. Whilst planning our experiments, we got curious about the properties of vesicles and if they could deliver cargo into plant cells. We figured it would be easier to test this with CMVs rather than OMVs, since CMVs contain all the contents of the cell and thus could serve as a delivery system for DNA, proteins, and other biomolecules. This would require a fusion of the plant membrane and the bacterial membrane, despite the plant's cell wall. But is that even possible? To find out, we tried using a non-amplifiable construct, but then soon realized this was like searching for a needle in a haystack. So we contacted Professor Olivier Wonnet, an RNA silencing expert at the ETH, about the possibilities for signal amplification using RNAi technology. He suggested to use a viral vector containing only GFP and a replicase, which will amplify the signal in a single cell by replicating itself. In addition, we used P19, an RNAi blocker, to inhibit the plant's antiviral response. So we infiltrated wild-type N-benthamiana with Agrobacterium as a positive control, E. coli bacteria, and CMVs from E. coli containing our plasmids. Since this viral vector is non-virulent, it can't spread systemically. So we knew even if a fusion happens, it would only be the case in isolated cells. Therefore, we looked at our plants under the confocal microscope. Here, you can see our results. This is the positive control. You see, the cells are jam-packed with GFP, which makes sense since Agrobacterium is an established DNA delivery system. And this is a picture of an epidermal cell from an E. coli infiltrated leaf. The GFP illuminates the nucleus and the cytosol, which are pressed to the cell borders by the vacuole. And here is the CMV infiltrated one. As you may notice, well, you see nothing, or at least no clear cell outlines. We weren't able to find evidence that CMVs can deliver DNA to plant cells, which doesn't necessarily mean that a fusion is impossible, but might be due to our mutagen damaging the viral vector. Nevertheless, our findings with the E. coli infiltration are interesting. Could E. coli possibly possess a delivery system allowing for the transfer of DNA between plant and E. coli cells? Well, we can't exactly answer this question at the moment. Our results could also stem from contamination, so we are conducting follow-up experiments to replicate our results as well as using a different mutagen for CMV production.
But for the moment, let's go back to the main part of our project, our engineered OMVs. Before we could even start with building the constructs, we needed to answer two preliminary questions before producing or even analyzing data regarding our constructs. So the first question: how does the OMV triggered immune response compare to the one triggered by CMVs? We tested the impact of the vesicles on the plant's immune system by means of a seedling growth inhibition experiment, or short SGI, where a lower seedling weight is an indicator for an immune response. The test showed that there is an impact of dosage on seedling growth, and the plots seem to show that lower concentrations of OMVs are required to induce an immune response than of CMVs. If the OMVs could sense when sprayed onto the plants, no large amounts would be needed, which would be another great advantage compared to commercial synthetic pesticides. And secondly, which strains vesicles trigger the highest immune response in plants and which receptors induce the response? Here, the treatments have a significant different effects depending on the plant's genotype, indicating that at least some of the response caused by membrane vesicles is dependent on the presence of certain PRRs. Post-hoc tests imply that the soil bacteria P. putida would be a good choice for setting bacteria free, because relatively to the others, their OMVs cause the strongest immune response. We investigated how adult plants react to vesicles with assays that quantify the reactive oxygen species burst, or short ROS, which is one of the first signs of pattern-triggered immunity. This suggests that treatments do have an effect, but there is no difference between the different plant genotypes in their overall immune response to treatments. The data suggests that the absence of the PRR EFR1 affects the immune response, which could imply that the immunogenicity of strains could be dependent on ELF18 or EFTU. To conclude, OMVs generally induce a higher immune response than CMVs. P. putida would be the adequate chassis if we were to release a bacterium into the wild. And with these two preliminary questions answered, we could test our own vesicles. By quantifying the lipids and proteins in isolated OMV samples from different E. coli strains, the tall B deletion strain is the best pick in comparison regarding the trade-off between growth rate and the quantified OMV production. We also chose the OMP8 deletion strain, in which all 8 outer membrane proteins are deleted, for a second type of construct because it elicited a remarkable immune response in previous experiments and has more space for the membrane proteins than tall B. Tol B was nevertheless crucial because our plan of an inducible system with the TEF protease depended on it. FLAG22 and ELF18 were chosen as elicitors to be built into the membrane with the aforementioned ClyA, because they are recognized as pathogen-associated molecular patterns, or short PAMPs, in many different plants. Data has shown that our constructs can elicit an immune response in Altaliana different from the induced ClyA only, which is a functional negative control that shows the immune response induced by the construct is not mediated by ClyA. From said data, we may hypothesize that first, OMP8 vesicles eliciting a higher immune response than tall B ones could be due to the fact that the OMP8 deletion strain has more space on its membrane because more ELF18 could be built into the membrane. And secondly, that ELF18 has a bigger impact than FLAG22 when used as an epitope. This would be in accordance with other findings and predictions that ELF18 is a stronger elicitor than FLAG22. These, however, are just two interesting hypotheses, and there could be other explanations for it. Furthermore, the SGI shows that the constructs are capable of inducing an immune response and that their concentration does matter. The post-hoc tests indicated that this difference becomes visible from 10 micrograms per milliliter. So, to sum up, generally, OMVs are more potent than CMVs, Peptida would be the adequate chassis for releasing, and our constructs work and are able to induce an immune response.
But even though our project works in the lab, what would a real-world application actually look like? People would need to be willing to use and buy our product. And in Switzerland, this is a bit tricky. We have a ban on the commercial use of GMOs, and Swiss farmers even decided voluntarily to renounce GMO-derived feed. This reflects a general distrust of GMO products in Switzerland. We found that surprising, since GMOs are already being used in other sectors, such as the medical and pharmacy industry. It turns out the term GMO-free is associated with good quality in Switzerland. But what does that even mean? Does it make sense? This is why we talk to scientists, politicians, as well as farmers to understand why people think this way. We came to the conclusion that if our product is to be successful outside of the lab, we need to strengthen education on the subject, prove that it is commercially feasible, and show that change is essential for a sustainable future in agriculture. So first, we talk to the ones who are actually involved in agriculture: farmers, companies, politicians, and scientists. Trying to cover the linguistic diversity of Switzerland, we had interviews in German, Italian, Swiss, German, and French. It turned out that the majority of the farmers were very enthusiastic about our project and would like to try it. We shared our interviews in a video series on YouTube. Later, we visited 3 high schools across 3 cantons in Switzerland, in which we introduced students to synthetic biology as well as our iGEM project. In addition, we held an event in collaboration with the Life Science Center to give high school students the opportunity of actually working with the same plasmids as the ones we are using in our experiments. It was important to us to reach as many demographics as possible. We further collaborated with the Swiss Study Foundation and organized a visit to the Agroscope, the Swiss Center of Excellence for Agricultural Research in Zurich. There, students from multiple backgrounds learned how we can analyze pesticide residues in our soil, why biosecurity research is crucial to us, and how field trials with GMOs work in the real world. In a second event, titled Pizza and Philosophy, we collaborated with the independent think tank REACH. Following our invitation, Professor Keller from the University of Zurich spoke about GMOs and his research on plant pathology, which later on enabled an evening of discussions on the ethics behind GMOs. We were again joined by a multitude of eager learners from fields outside of biology, from 18 to over 50 years old. To further understand the context in which BOOMV would need to persist, we decided to enter 2 collaborations, with EPFL as well as with NUS from Singapore and MIT from Manipal in India. There, we sought to find out what the consumer perception currently is around GMOs and pesticides. We gained valuable insights into the habits of farmers, producers, scientists, and consumers across these different continents with surprising results. They are now also available on YouTube. We observed similar key points in all 3 countries. Ease of use, price, and sustainability are the 3 key points to take away for farmers and producers, while education is most important for consumers. Finally, in order to show that alternatives to synthetic pesticides are needed if we want to move towards a more sustainable agriculture, we wrote a handbook. There, we analyzed the politics behind GMO regulations in Switzerland as well as in the European Union. We looked at the ethics of GMO use and made a commercial viability analysis. To summarize, we see that the status quo in agriculture is not where it should be if we strive towards a sustainable future. We need to develop alternatives to synthetic pesticides that are cheap to produce, easily available, and simple in their use. Also, the biggest factor in this race against the climate crisis is education, contributing to better informed consumers and a more democratic distribution of knowledge.